We'll be talking on chapter 6, a tour of the cell. So we need to know some learning objectives for this chapter, which are definition of a cell, how many types of cells are there, what is cell fractionation, the two types of electron microscopes, and the differences between plant and animal cells. So the first definition is a cell. A cell is defined as the basic, structural, and functional unit of life. And so we see that all cells come from a pre-existing cell according to the cell theory. Now we see that the cells cannot be seen with the naked eye. Therefore, we use sophisticated microscopes called electron microscopes. And electron microscopes are of two types: scanning electron microscopes and transmission electron microscope. So we see what is the difference between a transmission electron microscope called TEM or scanning electron microscope called SEM. Both these microscopes are going to use beams of electrons to view the image of the specimen so that we are able to see every detailed structure or the surface of that specimen or organism. Now, scanning electron microscope is focusing on the primary beam of electrons, and it provides 3D images of the surface of the different structures present in a cell. Whereas transmission electron microscope will show you every detailed structure of the internal organelles within a cell. Now we look at one technique which is called cell fractionation. So what's cell fractionation? To fractionate is to divide, to separate out. This technique or process called cell fractionation is going to take apart all the major organelles in a cell and separate them from one another. So we see, how can this be done? We can centrifuge tissue cells in a test tube. You mix them in a blender to create a homogenate. Once a homogenate is made, you subject the homogeneous mixture to centrifugal force. So you centrifuge it at high speeds. And so you can vary the speeds to identify different cell organelles from the tissue cells that you have homogenized. For example, if you spin it at 1000 G or 1000 revolutions per minute for 10 minutes, you get a pellet rich in nuclei and cellular debris formed at the bottom of the centrifuge tube. If you spin the same tissue cells at 20,000 for 20 minutes, the pellet will be rich in mitochondria and chloroplasts. If you spin it at 80,000 for 60 minutes, you can now separate the microsomes. And at 150,000 G or RPM for 3 hours, now you can separate the ribosomes. So depending on the time intervals for each of these cellular components or organelles at different speeds, you can now separate different organelles. So now we need to see what are the two main basic types of cells. We have prokaryotic cells and eukaryotic cells. Prokaryotic cells were the cells that lack a nucleus and membrane-bound organelles. And the only prokaryotic cell we are going to go into detail would be the bacteria. So bacteria and viruses are prokaryotes. Opposite of prokaryotes will be the eukaryotic cells. Eukaryotes will have a nucleus and membrane-bound organelles. Now, when we did taxonomy classification, all those living organisms were divided into three main domains: bacteria, archaea, and eukarya. So the bacteria and the archaea were the only prokaryotic organisms that we are going to look at. Protista, fungi, plantae, animalia are eukaryotes. So now let's look at the prokaryotic cell, namely bacteria. This is the structure of the bacterial cell. Now, bacterial cells, bacteria are microscopic. So the bacterial cell will only be seen with the help of a microscope, which we use in the labs, which are light microscopes. Bacteria, they have a cell wall. So they have two layers or two membranes, outer cell wall and this inner membrane, which is called the plasma membrane or the cell membrane. Now, plants have a cell wall, but the composition of the cell wall in plants is cellulose, whereas bacterial cell wall is made up of complex substance called peptidoglycan. So here they just call it glycocalyx. How do bacteria now enter into a living organism like us? It has these hook-like structures called fimbriae. Some authors call it pili. And with the aid of these fimbriae, they can now enter into us and cause the infection. So now we see, in order for an organism to be living, it must have DNA, the genetic material of the cell. But because prokaryotes don't have a nucleus, they don't have all these membrane-bound organelles, their DNA is not as complex as eukaryotic DNA. Their DNA is in the form of a single loop, which is present in a specialized compartment called the nucleoid region. So let's go now to what is the main features that all cells should possess. All of the cells should have a cell membrane. They should have cytosol, which is a semi-fluid substance. They should have chromosomes present, which carry the genetic information, or they carry the genes, and ribosomes, which will carry out protein synthesis. So we see now in eukaryotic cells, Protista, fungi, plantae, animalia, all these kingdoms belong to eukaryotes. What do we possess? We have a nucleus and membrane-bound organelles, and our DNA is present in the nucleus of a cell. We have cytoplasm. Cyto comes from the word cell, plasm comes from the word fluid. So cytoplasm is present between the plasma membrane, and cytoplasm is surrounding the nucleus of the cell. And normally eukaryotic cells are larger than the prokaryotes. Now we have this plasma membrane. What is the function then of the plasma membrane? It is going to permit small molecules in the form of ions or gases to go across it. Why? Because this plasma membrane is semi-permeable, or it is also said to be selectively permeable. So it's going to choose what it wants to come across it. Second function of the plasma membrane, it is going to separate the outer environment, the external environment, from the internal environment. And thirdly, it will allow substances to exit the cell that are not wanted or are in excess. So it is critical for every cell to maintain a surface area to volume ratio. So when we look at the cell now, we can see here, this is what our plasma membrane looks like. Our plasma membrane is a very intricate structure. We refer to it as the phospholipid bilayer, meaning it is our plasma membrane consists of two layers of phospholipids. The phospholipid head is hydrophilic because it faces water, and the hydrophobic fatty acid tails because they fear water. So these are water-loving molecules, water-loving heads, and water-fearing tails. We'll go into detail with the plasma membrane in the next chapter. So this is now the animal cell. And we're looking to see what is the difference between an animal cell. And if we look here, we want to see now, slide number 13 is the plant cell. If we look at the plant cell here, is we see that the nucleus of the cell normally will occupy the center, but in the plant cell, the nucleus is not in the center as in the animal cells like us. Why? Because the plant cells, they have a large central vacuole. That's the first difference. Whereas animal cells do not have a vacuole. Secondly, plant cells will have a double thick cell wall. So they have an outer layer called the cell wall, and they have the inner layer here called the plasma membrane. Whereas we animal cells only have a plasma membrane. Thirdly, plant cells, they have chloroplast present, whereas animal cells will not have chloroplast present. Now, animal cells, on the other hand, we have lysosomes present, whereas a plant cell does not have lysosomes. Animal cells, we have a centrosome, also referred to as centrioles, which are present, whereas plant cells do not have centrioles or centrosomes present. So these are the main differences in terms of organelles between an animal cell and plant cell. So now let's go to looking at the slide which is going to tell us about the nucleus of the cell. The moment you think the nucleus is called the control center of the cell. Why? Because it has all the information necessary to control the activities within the cell. And within the nucleus of the cell here, as we can see, when you look at the structure, the nucleus has a membrane called the nuclear envelope. And this nuclear envelope, it has openings which are called nuclear pores, like the pores on our skin. So what do these nuclear pores do for the nucleus? They allow substances to enter the nucleus. It's the nuclear envelope having these openings called the nuclear pores. Now, within the nucleus, you have a small rounded body, which is called the nucleolus. And the nucleolus is going to help to carry out protein synthesis. Now, in this whole nucleus, you have these thread-like structures called chromatin. Chroma is color in Latin, tin is threads. So the moment chromatin is present within the nucleus of a cell, you know that the cell has not begun cell division called mitosis. Now, what happens to this chromatin? Chromatin, which is going to coil and condense with the help of histone proteins to form chromosomes. So the moment you see chromosomes within the nucleus of a cell, immediately you have to associate it with mitosis, cell division. That is an indicator that cells have begun dividing. So the two keywords are chromatin and chromosomes. When you see chromatin within a cell, the cell has not begun mitosis, cell division. Chromosomes are visible, mitosis has occurred. Let's go now to seeing what then would be ribosomes. So when you look at ribosomes, where are we seeing ribosomes? They are the protein factories. But we want to see where are these ribosomes present within the cell. So we're going to see the structure of these ribosomes and see where exactly ribosomes will be present within the cell. Ribosomes are nothing else but it is the rough endoplasmic reticulum. ER, the acronym ER refers to endoplasmic reticulum. And endoplasmic reticulum is of two types, rough ER or smooth ER. Now, why would it be referred to as rough ER? Because ribosomes are present on the outer surface of the cell, making it rough. And I want you to connect these words when you are studying. So when you see rough ER is so called because the outer surface is rough because ribosomes are present. So rough ER, ribosomes present, immediately connect the function to protein synthesis. Therefore, ribosomes, they are referred to as the factories of the cell because the moment you see ribosomes, it is the site or location where proteins would be synthesized. Now, what would be smooth ER? What is smooth ER? No ribosomes are present. Therefore, the surface is smooth. And immediately, the connect or associate the function to be lipid, to carry out lipid and fat synthesis as well as carbohydrate synthesis. So this is the importance of the endoplasmic reticulum, the two types which are rough ER and smooth ER. Now we talk about the next slide, namely Golgi apparatus. So what is the function of the Golgi apparatus? First, we must know what does the Golgi apparatus look like. So structure, function, location in a cell is very important. Now, all this information I've also added in my handout called Cell Organelles Notes. So if you look at Golgi apparatus, the Golgi apparatus is collectively also called Golgi complex. What does this Golgi apparatus consist of? It consists of these vesicles called cisterna. All the vesicles are cisternae. Cisterna is one. Each of these vesicles called cisternae are also referred to as Golgi bodies. In totality, altogether, the cisternae or the Golgi bodies will form the Golgi complex or the Golgi apparatus. Now, the Golgi apparatus is going to perform four functions. It's going to collect, it's going to modify, it's going to package, and then it's going to distribute or sort. So, as we see in the Golgi complex, you have two sides. You have a cis side, which is the receiving side of the Golgi apparatus. And then you have the transport side, which is the shipping side called the trans side. So cis is receiving and trans is shipping or sorting out. This is the TEM of the Golgi apparatus. Let's go to now lysosomes. Lysosomes is a typical animal cell organelle. Now, lysosomes, they are membranous sac, meaning they're protected or enclosed by a membrane. And what do these lysosomes contain? They contain the digestive enzymes in our bodies, which are all typically called hydrolytic enzymes. So these enzymes, they are going to digest the worn out red blood cells, white blood cells in the body, which are causing the infection. And so what happened when we were infants, why don't we have a tail? It's because the lysosome secreted these hydrolytic enzymes which ate the cartilage of the tailbone. And what are we left with now? The rudimentary appendage, which we now call tailbone. That's why we are not born with tails. It's the lysosomes that have done this function. And the lysosomes, they work best in an acidic environment. Now, we see the process of phagocytosis. Phagocytosis comes from the word phagein, phagophagein in Greek, which means to eat. So when the cells are engulfing or eating a large solid molecule, like a food molecule, or even if it engulfs an entire microorganism like bacteria or virus, and the vesicle is now going to close once it traps it, and then the digestive enzymes are going to be released and break down those food molecules or digest the entire microorganism and then release it. So recycling of the cell's organelles is being carried out with the help of these hydrolytic enzymes that are being released by the lysosomes. Now, lysosomes are also going to recycle these cells' own organelles through a process what is called autophagy. So we can see how autophagy takes place in the lysosome or phagocytosis that I just explained. Now, if we go to a vacuole, we know plant cells have this large central vacuole. What are vacuoles? Vacuoles, they are large vesicles. They can be derived both from Golgi apparatus as well as from the endoplasmic reticulum. But you have other types of vacuoles in lower organisms like in bacteria. You have food vacuoles. We have contractile vacuoles like in amoeba and other protists. What are the food vacuoles going to do? Food vacuoles, they are going to engulf the food and the vesicle is going to close so that the food gets trapped through the process, as I just explained, by phagocytosis. Contractile vacuoles, as we see in amoeba, amoeba is a freshwater protist. And what does this contractile vacuole do? It is going to expel out all the excess of the water from the cell. And plant cells, which have the large central vacuole, they are going to store all the organic compounds as well as water. So nutrients and organic compounds would be stored in the large central vacuole in plant cells. So now let's go to the next slide and we're going to see what is mitochondria. Mitochondria and chloroplast, pay careful attention. A number of questions will be asked. The moment you think of mitochondria, mitochondria is called the powerhouse of the cell. Why is it called the powerhouse of the cell? Because it makes energy. It's also referred to as the energy currency of the cell. It's making energy in the form of ATP, which is adenosine triphosphate. And from the last previous chapter, I explained to you in the recording what ATP is, how adenosine triphosphate gets broken down to adenosine diphosphate and adenosine monophosphate. Once the phosphate organic bonds are broken down, they get converted into PI, which was inorganic phosphate. So what is the process that occurs in mitochondria? Cellular respiration. When you look at cellular respiration, we need to know what then is cellular respiration. Cellular respiration is also referred to as oxidative metabolism. Why? Because oxygen is required by the cells and by the organisms to breathe. And so when you look at cellular respiration, we see that Anywhere between 36 to 38 ATP molecules are required by the cell for the cell to undergo cellular respiration. I want to show you here in this next slide here. What is the structure of mitochondria? Where exactly in the mitochondria does cellular respiration occur? So mitochondria consists of these two membranes, this outer membrane and the inner membrane. I describe mitochondria as this oval sausage-shaped structure. Now this inner membrane has these folds which are called cristae. What is the function of this cristae? It is to absorb the nutrients that are entering into the mitochondria. Now, the space within the inner membrane is referred to as matrix. And what do we? Matrix is the space. It is here in the matrix that cellular respiration is going to take place within a cell. And within this matrix, we can see there are free ribosomes that are present in the mitochondrial matrix. And all of us students, we inherit the mitochondria from our mothers, the maternal mitochondrial, maternal mitochondria, which is so crucial. So with this, we finish mitochondria cellular respiration. But when you look at cellular respiration, we need to know that there are two types, aerobic respiration and anaerobic. Aerobic comes from the word air, oxygen. So those organisms that require oxygen to breathe are called aerobes. We are all aerobes. Fish, humans. Now plants also require oxygen to breathe. Let us look at anaerobes. Anaerobic respiration is the presence of carbon dioxide gas and not oxygen. So anaerobes are organisms that require carbon dioxide gas to breathe, and it will be very toxic in the presence of oxygen. So who undergoes anaerobic respiration? We learned the only unicellular fungi was yeast. It carries out fermentation. Fermentation is a classic example of anaerobic respiration. And I want you to look into glycolysis, the breakdown of glucose sugar, C6H12O6 during glycolysis, which is one part that occurs, one process in cellular respiration. This is a TM of the mitochondria. So we've seen the importance of mitochondria. Now, the last organelle that we're going to talk about would be chloroplast. Chloroplast is a typical animal cell, plant cell organelle. And so when you look at chloroplast, let's look at the structure here first. It's a 3D image. And we see you have an outer membrane and an inner membrane. Now, within the inner membrane, you have these flattened discs. They represent like pancakes. You don't eat one pancake, you eat a stack of pancakes. Each disc is referred to as a thylakoid. And a stack of these discs are referred to as granum. Plural is grana, several stacks. But these thylakoids are connected to other thylakoids. So the grana are connected to each other through these structures, which are called lamellae. They haven't shown it to you in the diagram. Now, within the space in the chloroplast, you have this semi-fluid with granules, which is called stroma. Now, where does photosynthesis take place? Photosynthesis takes place in the thylakoids. Why? Because the thylakoids contain the green pigment called chlorophyll. That's why they are green in color, which is an indicator that photosynthesis is taking place there. So what then is photosynthesis? Photosynthesis is the process where green plants, they are going to manufacture their food with the help of the green pigment called chlorophyll in the presence of sunlight, carbon dioxide, and water. And oxygen gas is released. Glucose molecule is the energy provider of the cell. So now here, I want to just talk about this most important organelle which we need to know, which is the centriole. Okay, when you looked at the centriole, take note of this, that when we talk mitosis and meiosis, the centriole is the marker for you to know that this is a plant slide or an animal cell, because it's a typical animal cell organelle. Now, the centriole is made up of these microtubules. So we come to the part of the next structure, next organelle called cytoskeleton. Cyto comes from the word cell, skeleton is framework. What does our cytoskeleton consist of? Our cytoskeleton consists of three structures: microfilaments, intermediate filaments, and microtubules. Now we can see here these microtubules. So the way I distinguish it is micro is small, tubule is cylindrical. So the diameter of a microtubule is largest, microfilament is thinnest, and intermediate filaments would be, the diameter would be the size in between microfilaments and microtubules. I have explained in detail this, I have written the notes in detail for microfilaments, microtubule, etc. Please go through the handout there. And so this is the function of the cytoskeleton, is to give us the cells in our body shape and support. And so centrioles, you see, they would be forming, or centrosomes will be formed with the help of these microtubules. So you'll have 9 microtubules in a spherical pattern. And if you have 9 all around and nothing in the center, we call it a 9+0 arrangement. If you have 9 microtubules in a semicircle and 2 in the center, it would be a 9+2 arrangement, as you see it in cilia or in basal bodies. So with this, students, we have finished the presentation of the tour of the cell. I want you to listen to this lecture presentation, go through the detailed notes that I have given in the handout. Have a great one, students. Till next time, bye-bye.